Hello, friends. Welcome to my YouTube channel, The Microbiology Digest. I am Dr. DP Bora. Today, we will be discussing one of the very important topic of virology, which is required to be understood very well if we go deeper into the virology. That is the replication of virus, how virus replicates. So, this will be the discuss topic of today's discussion. So let's start. So first, let us understand what is actually the replication of virus. What we understand. So it is actually a process by which viruses they make copy of its own. Means virus will synthesize its own copy by the process of replication. All the viruses they must undergo this process of replication in order to create new or progeny infectious variants so that these variants will be able to infect new cells or other cells or other hosts. Once a virus enters to a body of a host, it makes a physical contact and tries to cross the plasma membrane of a target cell. So this is the first step in the replication process of viruses. So once, as we all know, the viruses, if they are outside the host cell, they behave like inert particle. And once they come in contact with a host cell, so first they will try to make physical contact and they will try to cross the membranes, plasma membranes of the cell so that they can enter to the cell. Once they enters, they will try to they will try to release its genome, which is carried by the virus, and the genome will be replicating. And along with this replication process, the virus will try to manufacture its proteins in different locations of a cell with the help of the ribosomes of the host cell. Then these virus particles, they will be assembled, and finally, once assembly is complete, they will be released from the cell, and they will continue the process of infection by infecting a new cell. So this is the entire process of virus replication in a nutshell. So this genetic information which is carried by the virus or viral genome that is required for replication of the virus, it is carried by the virus or carried in the viral genome, and they will have to take the help of host cell machinery because viruses, they, as we all know, they lack the biosynthetic enzymes, biosynthetic machinery. Viruses, they do not have these machineries, and that is why they are dependent always on the host cell. So, in simple words, when we say replication of virus, it means the process by which virus will make its own copy or synthesis of new variants. So that is the process of replication of virus. Now, what are the different steps in viral replication? So viral replication, it is composed of 7 distinct steps, but many a times in some viruses, these steps may not be clear-cut divisions, or they may occur as overlapping stages. So these steps are starting from the beginning, it is attachment, penetration, uncoating, replication, assembly, maturation, and release. These are the 7 steps of virus replication. These steps can be serially, there is one easy way to remember these steps by one line that a purple apple might redden. So these red letters marked in red color, they represents the different steps or first letter of the steps of virus replication. So this is one of the easy way to remember the different various stages of virus replication. So all viruses, they must undergo these 7 steps in order to complete the replication process. So now we'll be discussing every steps one by one. So starting with the attachment step. This is the first step in the virus replication process. The virus, when it comes in contact with a cell, new cell, first, as in the first very beginning, we discussed about that virus will try to establish physical contact. That is what actually the attachment step is. This attachment, it is nothing but the binding of the virus to a host cell. This is highly specific process. This to complete this process, virus, they carry some attachment proteins which helps the virus to attach to the cell surface. Okay, so these attachment proteins will be adsorbed to the receptors which are present on the host cell. So there will be highly specific binding between the attachment proteins in the virus and the receptors present in the host cell. And these molecules, they are present in different cells which will be very well recognized by the virus. And these receptors, they are usually glycoproteins, or they may be carbohydrate residues. They are present in the plasma membrane through which virus will make physical contact or attachment so that the replication process can be initiated by the virus. So these are some of the examples of the receptors by which viruses, they established physical contact with the host cells. There are different types of receptors has been identified and which are used by different types of viruses. Next step in virus replication is the penetration. After attachment, the viruses, the viruses which are successful in establishing a successful contact or attachment with the virus with the host cell, they will try to enter to the inside of the host cell. This penetration, actually, it refers to the crossing of the plasma membrane by the virus so that virus can enter to the interior of the host cell. So different mechanisms are utilized by the viruses to gain entry. One of one of the mechanism is the endocytosis. This occurs through Clathrin coated pits, and these pits fuses with the lysosomal membrane, and then lysosomal degradation, and finally there will be release of the nucleic acid. This happens mostly in case of influenza viruses. And receptor mediated endocytosis, it is one of the very common method which is used by majority of the viruses to penetrate the plasma membrane. So what is that method? That is receptor mediated endocytosis. Another method of penetration is the fusion. Here what happens, the mostly in case of envelope viruses, this method of penetration is utilized. Here the viral envelope, they can fuse with the cell membrane, and this fusion will lead to formation of an endosome through which the virus will gain entry to the host cell. The third method is by translocation. This is another method by which mostly the naked icosahedral viruses or icosahedral viruses which do not carry any envelope, they mostly utilize this method of translocation to gain entry to the host cell. Third method of virus replication is uncoating. So uncoating, it refers to the breakdown or removal of the capsid. So once viruses, they enter successfully to the inside of the host cell, so they will have to release the genetic material carried by the virus to the internal environment of the host cell. For this, they needs to break the capsid. So this uncoating, actually, it is the process by which capsid are removed and the viral genome is released to the cell so that the genome can undergo replication as well as transcription of this genome will take place. So this is uncoating. So depending on the viruses, uncoating may occur at the cell surface, or it may occur within the cytoplasm, it may occur at the nuclear pore, or it may occur within the nucleus. So depending on the type of virus, the place of uncoating may differ. So depending on the site of replication, the virus will undergo uncoating in the preferred site where the virus prefers to replicate. Fourth step is the replication. So this is the major step. We can say this step in some cases it may be referred to as viral macromolecular synthesis. So replication, it is the making a copy of the viral genome, and this genome, the instruction which are required to undergo this step, it is carried in the viral genome itself. So this, as we all know, viral genome, it codes for different types of proteins. Okay, so to synthesize new virions, first, the virus has to synthesize these new proteins. So this synthesis of new proteins, it comprises the step of replication. So in order to replication to occur, initially two events are required to complete. First is the transcription of the early mRNA and translation of the early proteins. These early proteins, they include mostly the enzymes which are required for the replication of the viral nucleic acid. Okay, the early proteins, these includes mostly the enzymes required for viral replication. Finally, once the replication has initiated, then it will be completed after these two events are over. First is the transcription of the late mRNA and translation of the late proteins. So once these two events are completed, then replication of the viral genome is also complete. So these are the replication strategies depending on the virus, type of virus. They employ different type of strategies, and these strategies are illustrated here. These are based on the Baltimore class, as we have already discussed. Baltimore classification classifies viruses into 7 different classes based on the strategies employed by the viruses to undergo replication. So class 1, class 2, class 3, they all these viruses, they carry different type of nucleic acid, and based on the type of nucleic acid, they employ different strategies to undergo replication. Fifth step in the virus replication is the assembly. Virions, they are, or viruses, they are created from the newly synthesized components. So what we have discussed so far, once virus nucleic acid undergoes replication, so different viral components are synthesized in different locations. So these different viral components are assembled to synthesize a new virus particle. So this synthesis of new virus particle by assembling the different components synthesized newly at a particular site, this entire process is called as assembly step. So location of this step, assembly step, it will depend on the type of virus. It may take place in the cytoplasm, it may take place in the nucleus itself, and some viruses, they assembly may takes place in the Golgi complex. So depending on the type of viruses, as site of assembly may differ. Once the viruses are assembled, new viruses are assembled, these viruses, they are formed as an immature virus particle. Immature virus particle, and once they are completely assembled and released from the cell, then they become a mature virus particle, which are also otherwise called as virion, and they are capable of infecting a new cell. Sixth step is the maturation. Maturation, it refers to the final changes within an immature virion which were assembled, and the maturation step results in synthesis of an infectious virus particle. So during this step, structural changes will be occurring, capsid changes will occur. Then these are mediated mostly by the host enzymes or virus-coded enzymes. And once maturation is complete, a completely mature virus particle will be ready, and this will be ready for release from the cell. And final step is the release of the virus. So this is the final step in the replication process, and this involves the release of the newly synthesized virus particles from the cell, and this will enable the newly synthesized virus particle to infect the new susceptible cell. And release of the virus, it mainly takes place by two major mechanisms. First mechanism is through budding. This is the process which is used mostly by the envelope viruses. All the envelope viruses, they are released from the cell by the process of budding. Then second method is the by cell lysis. So here non-envelope viruses, they are released from the cell by the process of cell lysis. So cell undergo lysis and virions are released from these cells. So these are the different steps of virus replication. So what are the steps? Initial, starting with the attachment, then penetration, then uncoating, replication, maturation, assembly, and finally the release. So these are the 7 stages of virus replication. Next, one very important experiment conducted to understand the virus replication process by two scientists, Ellis and Delbruck. So this is called as one-step growth curve. So one-step growth curves, they are used to study the virus replication cycle. So replication cycle of a virus is studied with the help of one-step growth curve. So one-step growth curve, the name it is given because the viruses replicated simultaneously, and all the events they are in one step. That is why the entire experiment is termed as one-step growth curve. So here one term is used that is called as multiplicity of infection. In short, it is called as MOI. It refers to the ratio of virus to the cells used for infection. So one MOI, an MOI of 1 means one virus particle is used per cell, and 10 MOI means 10 virus particles are used per cell to study the replication cycle. So generally, when we conduct one-step growth curve experiments, a high MOI is used so that it ensures the infection of all the cells by the virus. So this is one-step growth curve. What happens here? In this type of experiments, cells are infected with a virus at the same time, and the increase in the infectious virus over time is followed by sampling and titration of the virus at different time intervals. So this is how one-step growth curve experiments are conducted. So this is a typical curve which is generated after one-step growth curve experiments. So typically this has 4 different stages, phases. First phase is the infection. It includes the attachment and penetration phase. Then second is the eclipse phase. So in this phase, no virus, infectious virus could be detected. And during the eclipse phase, the virus enters to the cell, and there's a latent period. It is entry to release of the virus to the medium, and fourth phase is the logarithm phase, or this is the production phase. So newly formed viruses particles will be released from the cell. These are the typical phases of a one-step growth curve experiment. So eclipse phase, as we have discussed, shortly after infection, what happens? Inoculated virus, it gets disappeared from the medium, and no virus can be detected. Okay, and this phase is called as eclipse phase or eclipse period, and it the length or duration of eclipse period, it differs from virus to virus, but generally it ranges from 2 to 12 hours. Okay. And in one step growth curve or virus replication, one term is used that is called as burst size. So what is burst size? It is the average number of virus particles produced per infected cell. Average number of virus particles produced per infected cell. It denotes the burst size of a virus replication. So this is all about virus replication. So I hope this information will be helpful, and please subscribe to my channel if you feel that these are helpful, and give your comments. And with this, thank you for today.